Hello, my name is Ana Paulina Cardona and I am going to talk about apolipoprotein E imbalance in the cerebrospinal fluid of Alzheimer's disease patients. Alzheimer's disease is a progressive neurologic disorder or a type of dementia that atrophies the brain, and age here is the main risk factor. This disease affects memory, thinking, and behavior. On the other hand, we have apolipoprotein E, which is a proteic component of lipoprotein particles that carry cholesterol in the blood. Apo E is in the plasma as well as in the cerebrospinal fluid. And in humans, there are three versions of Apo E gene that are E2, E3, and E4. And each additional copy of the Apo E4 allele is associated with a higher risk of having Alzheimer's disease. The purpose of this study was to examine the levels of cerebrospinal fluid apolipoprotein E species in Alzheimer's disease patients. We have four methods. The first one is ELISA, that is a test used to detect antibodies or antigens in the blood. So the antibody or the antigen that is going to be studied binds to a specific enzyme in order to detect it. And it was used in this study to measure the levels of the Alzheimer's disease biomarkers, because according to those results, they were classified as AD patients or controls. We also have transgenic rat CSF. These rats are animals that express some human genes, and they were used in this study to collect CSF samples, because they also evaluated different proteins that included brain analysis in each stage, and these, of course, couldn't be carried out in humans. Western blot is a technique used to detect a specific protein. It involves using electrophoresis to separate proteins and then exposing them to a specific antibody against the protein under study. It was used since the presence of Apo E had to be evaluated, and as it is a protein, it was going to respond to this technique. The last one is immunoprecipitation. This is a technique in which a protein antigen is precipitated from a solution using an antibody that binds to that protein. And this procedure was used to isolate and concentrate the protein, that in this case is Apo E. Talking about the results, here in the figure 1, we are analyzing the CSF obtained from wild rats or transgenic rats at different ages. And as we can see, only at 16.5 months of age, Apo E levels were higher in transgenic animals than in wild types. In the figure 2, we have the results of human patients. First of all, in all CSF samples, Apo E appeared as two distinct bands of 34 and 36 kilodaltons. We can also see a band of 100 kilodaltons, but only in the samples of AD patients. And specifically here in the figure D, we have heat and two deglycosylating enzymes. And the glycosylation didn't alter the Apo E band pattern, but O deglycosylation simplified the Apo E pattern to a single band. Here in the figure 3, they used an electrophoretic technique that separates proteins according to size and charge. So they included a CSF control sample with denaturating conditions as well as an Apo E3 recombinant protein under native conditions. And this helped to see the Apo E monomers and dimers thanks to the discrepancy in the molecular weight. In the discussion, the first thing that the author said is that even though some previous studies didn't distinguish the Apo E species, have indicated that CSF Apo E levels in Alzheimer's disease patients are increased, and this completely agrees with the article because they proved Apo E levels increased in those patients. The other authors mentioned that the 34 and 36 kilodalton species are likely different of glycoforms, and the difference in electrophoretic mobility of the Apo E glycoforms could be a consequence of its sialylation. This also agrees, and it is even related to figure 2. And the last one indicates that Apo E is exclusively glycosylated and can be capped with 1 or 2 sialic acids. It is true, and it is related as well with the figure 2. Finally, we have the conclusions. The first one is that molecular biology influences medicine as it deals with understanding and treating the specific details that cause each of the pathologies. Besides, it gives us the basis for disease prevention through research such as the one that was carried out in this article, given that if we discover the molecular basis of a disease, we will have tools to avoid it, and we will have also more tools to overcome the disease. Thank you for your attention. Goodbye.